Hello, everyone. My name is Stephen Wang. I'm an assistant professor of genetics and cell biology at Yale. Today, I'm kind of talking about our work on the multiplex imaging of nucleome architectures in single cells of mammalian tissue. We know the interface chromosome is spatially organized in a mammalian cell. On the first level, DNA wraps around histones to form these nucleosomes or 10 nanometer fiber structure. And then on the other end of the spectrum, we actually have individual chromosomes occupy distinct nuclear space, forming these chromosome territory structures during the interface of cell cycle. And in recent years, thanks to the very powerful, very beautiful high-C technique, we now know that at the length scale of several hundred kb, there are these topologically associating domains, also known as contact domains. These are continuous sections on the genome that are making a lot of self-contact. And within the TAD, there are often chromatin loops, such as promoter enhancer loops, and sometimes the whole TAD itself is a giant loop itself. And these TADs are considered building blocks to build up these chromosome territories. And when they do so, they seem to be segregated into two compartments called the AB compartments. Other beautiful sequencing techniques also profiled regions of the genome that are associating with the nuclear laminar and associating with the nucleolus. And these are termed laminar associated domains, or LADs, or nucleolus associated domains, or NADs. And we now know that all these levels of genomic architectures or nucleomic architectures, they play an important role in multiple genomic functions, especially gene expression regulation. For example, we know that the TADs largely define the scope of promoter enhancer interactions. And the AB compartments, the A is enriched with active chromatin and B is enriched with inactive chromatin. The LADs and NADs are enriched with inactive chromatin also. However, despite all these beautiful advances, there are still a lot of unknowns in this field, largely due to technical limitations. And there are two groups of techniques in this research: imaging-based technique and sequencing-based technique. Among the imaging-based technique, definitely, I think the FISH, or fluorescence in situ hybridization, is a very representative one. And in conventional DNA FISH, what we have are these oligonucleotide probes that are labeled with fluorescent dyes. And upon heat denaturation of your genomic DNA, they can hybridize to the genomic target. And then one can use fluorescent imaging to image all these targeted and labeled genomic loci. Because it's an imaging-based technique, you immediately see the real 3D positions of all these labeled genomic loci, and it's very easy to look at single cells. However, the challenge of doing FISH is usually it only detects a few genomic loci at a time. That's because one needs to use different fluorescent colors to distinguish the different loci, and there are only a few spectrally distinguishable fluorescent colors available for fluorescence microscopy. So this is not really a systematic approach. And on the other hand, among the sequencing-based technique, high-C is very well known, and it's biochemistry and sequencing-based technique that measure the contact frequency among pairs of genomic loci. It's a really systematic technique. You get pairwise contact frequency among the whole genome, but it's not really looking at the real 3D positions of all these loci. And another thing is, it usually depends on population averaging to get high enough genomic resolution. There are single cell versions of high-C available, and they are becoming more and more powerful. But up to this point, the majority of the high-C discoveries, the conclusions are based on population averaged analysis. So we see a key technical challenge here is how do we get real 3D position measurements in the single cells at the systematic level, right? So FISH gives us 3D positions and in single cell, but not really systematic. High-C don't really give real 3D positions. It's rather contact frequency. It is systematic, and it usually requires population averaging. So how do we get all three of these benefits? And because of this technical limitation, there is one key biological question that have haunted us for decades. That is, what is a 3D folding path of chromatin at length scales above the nucleosomes? Right? So from TADs or contact domains to the AB compartments to the chromosome territory, these are essentially larger and larger chromatin blobs. And we don't really know as in, for example, structural study of protein structure, we know the peptide folding conformation. For chromatin, we don't know that. We don't know what is the shredding folding path of chromatin, really. To address this question, during my postdoctoral work at Dr. Xiaowei Zhang's lab, I developed a technique termed chromatin tracing. And this is one technique to directly trace the spatial organization of numerous genomic regions in individual chromosomes in single cells. And in that work, we applied it to human cell culture. And to perform this chromatin tracing, the basic idea is that we want to label and image numerous genomic loci along the whole chromosome, and we want to link them one after another to reveal the chromatin trace. However, a challenge here is that if you simply label so many genomic loci along the same chromosome with the same fluorescent color, you don't know how to link which one with which one, right? You don't see the order of them in this map. And beyond this, there's another challenge, which is in reality, the fluorescence from all these genomic loci, they're all going to be connected due to the diffraction limit of light, like shown here. This is actually one chromosome, and you don't see all the labeled genomic loci in it. So we cannot even resolve so many different genomic loci when they're simultaneously labeled with the same color. To tackle this challenge, we developed this sequential imaging strategy. So what we do is we first label and image one genomic locus. And because this is one locus, it forms a diffraction limited spot. And we can fit its center position with much higher accuracy all the way to the nanoscale. So it's much more precise than the diffraction limited resolution of light microscopy. It's the same principle as used in single molecule based super resolution imaging like PALM or STORM, although we're doing conventional imaging. So we can fit its center position with very high accuracy. And then we can photo bleach this genomic locus and label the next genomic locus on the genomic map. And we can link their center positions because we know by design they are next to each other on the genomic map. And then we can bleach this locus again, image the next locus on the genomic map, link the bleach, and we do this procedure over and over and again until we image all the genomic loci of interest, and we can link their positions into this 3D folding path. So to realize this design, we developed this technique based on multiplex sequential fish. And here to trace all 34 tags along human chromosome 21, what we do is we first label all the tags with a library of oligonucleotide probes, labeling all the center 100 kb, all the center 100 kb positions of all the 34 tags. And at this stage, if we do a 3D fluorescence imaging, we see these two patches of fluorescence on top of the nuclear background, and this corresponds to the two copies of chromosome 21s in this diploid human lung fibroblast IMR90 cell line. And after photo bleaching, we can actually apply two secondary probes that binds to the overhangs on top of the primary probes associated with two tags only. And with 3D fluorescence imaging, we can image these two spots that corresponds to the center positions of these two tags. We fit their center position with nanoscale accuracy, and then we photo bleach and we image the next two tags. So this way the tags are imaged two at a time until all 34 of them are sequentially imaged with two color imaging. And then we can basically trace this 3D folding path of this very chromosome. So in this initial technique, we essentially profiled the chromatin folding at the tag to AB compartment to whole chromosome kind of length scale. After this work, my postdoctoral lab and several other labs, including Alistair, who just gave a talk 2 weeks ago, increased the genomic resolution of this technique by labeling finer genomic regions and traced finer structures such as tag boundary or promoter enhancer interaction, and expanded, and expanded the application of the technique from mammalian cell culture to also Drosophila embryos. And in our initial work, applying this to the tag to chromosome scale in human cell culture, we applied it to answer several specific biological questions. One of them is about the AB compartment organization. So at that point, people actually don't know whether the AB compartments are really stable physical structures within individual copies of chromosome, or are they just a population averaging trend, a statistical trend due to the population averaging in the high-C procedure? So if this is the chromosome and all the compartment B tags are labeled with as blue spheres and all the compartment A tags are labeled as red, high-C basically show that there is a higher contact frequency among the B tags and there are higher contact frequency among the A tags. However, is it possible that, for example, maybe in individual copies of chromosomes, you have transient interaction among pairs of A and pairs of B, and if you average this with many other copies of chromosome, it looks like all the A's are coming together and all the B's are coming together, but in reality, individual copies of chromosome is never the case. And on the other hand, if the AB compartments do stably exist, how are they spatially positioned? Is it polarized, like in a copy of chromosome, all the A are on one side and all the B are on the other side? Or is it radial, maybe A wrapping around B? In fact, this is one model that the several paper proposed. And we didn't know at that point. However, with our data, we can immediately see that. But to do that, we performed this compartment analysis following the computational workflow that was introduced in the initial high-C work. I won't go into the details here, but just show you that in the end, what you get is this compartment score plot. So on the x-axis, you have 34 tags, and these are the ID number of the tags. And if a tag is in compartment B, it's shown as blue bar here, and it has a negative compartment score. And if a tag is in compartment A, it has this positive red compartment score. And all of these are still based on population average analysis. However, with this AB compartment assignment, we can go back to the individual chromatin traces and ask, where is the A, where's the B? And here are two individual copies of chromosome 21 traces. And all the B tags are colored in blue and A are colored in red again. And we see immediately from these two copies of chromosome 21s that the AB compartments are really stably existing in individual copies of chromosomes, and they are positioned in this polarized side-by-side kind of fashion. And we further defined these convex hull shapes that enclose all the B and A, and we defined this quantity called the polarization index, so that if the two compartments, they are really separated and segregated, polarized in space, then it's the polarization index is 1. If one perfectly overlap the other or wrap around the other, the polarization index is going to be 0, and the partial overlap would be something in between. And we apply this metric to all the individual chromosome traces, and we find that for chromosome 21, the polarization index are often very high. Many of them are actually exactly 1, meaning that the A and B convex compartments are totally not touching each other, and the polarization indices are much higher than those from a control where the compartment scores are randomized, compartment assignments are randomized. And we saw the same thing for two other autosomes we studied, and we conclude that AB compartments stably exist in individual chromosomes and are organized in a polarized fashion. The chromatin tracing technique not only has single cell resolution, it actually has single chromosome resolution. So if your two copies of the same chromosome in the diploid cell actually differ from each other, we can actually see that too. And in the first work, we actually showed this with a study of the X inactivation. So we know that in female mammalian cell, the two X chromosomes are actually different. One of them is called the inactive X or XI, and the genes on it is largely transcriptionally silenced. And this is one way to deal with the dosage difference of X chromosome between a male and a female. And the XI, people know that it's more spatially compact. And with our technique, we looked further into how the chromatin fold and compartmentalize within XI versus XA, the active X chromosome. And we do that by co-immunofluorescence labeling, targeting the microhistone H2A histone variant that's enriched on the inactive X chromosome. And we can really distinguish in our fish images which X is XA and which X is XI. We can analyze their chromatin traces separately. Here I'm plotting the mean spatial distances between pairs of genomic loci on XI versus XA. And here I'm plotting the mean spatial distance versus the corresponding genomic distance between the two probed genomic loci. And we see that Xi and Xa indeed differ quite a bit on these plots. And these plots are actually showing that the Xi chromatin folding not only is more compact, it's actually more intermixed. It's more intermixed. So to the point that it doesn't even look like a polymer to a large extent. It looks like just randomly mixed particles. And then when we look at the compartmentalization, the Xi and Xa are also different. So Xi form these two large compartments separated by the DXC4 microsatellite sequence, and Xa form two compartments that correspond to the P and Q arms of the X chromosome. And these X-specific compartments, they are also stable existing individual copies of chromosome and are polarized. So we conclude that active and inactive X chromosomes adopt drastically different folding and compartmentalization schemes. And then we realize the chromatin tracing not only offers us direct visualization of 3D folding, and it's at the single cell level and it's at single chromosome level. There are several other advantages too. It should be compatible with co-RNA fish, and we show this compatible with co-immunofluorescence. So we have a chance to really combine multiple imaging modalities together to look at the DNA, RNA, protein all together on the same cell, on the same chromosome at a very multiplexed scale. And the chromatin tracing is expected to be broadly applicable to multiple genomic length scales, to different cell types and cell states in both cell cultures and tissue sections from different model organisms. And this is about exactly what my lab did in the past couple of years after I opened my lab at Yale as part of our research program. So recently, we introduced a new technology called the MINA. It's an integratory technique that allows us to profile multi-scale chromatin folding, other nuclear architectures, such as the laminar and nucleolar association, and the numerous RNA species in the same single cells in tissue. We used the mouse fetal liver tissue section, and we were able to analyze the chromatin folding and the nuclear architecture in a cell type specific manner with this capacity. So we developed this MINA technique with several biological questions in mind again. The first of them is in the native tissue environment, how is the genome folded in 3D across different length scales in single cells of different cell types, right? Especially for tissues like fetal liver, it's a tissue that is with mixed cell types. The cell types are not spatially very well organized, and the cells, their morphology looks quite the same also. How do you study chromatin organization in such a situation in a native tissue context? And secondly, how is the 3D genome folded in relation to other nuclear components, right? So we have the AB compartments, for example, but we also have LADs and NADs, so laminar-associated and nucleolar-associated domains. What's their relationship in all these different cell types? Previously, different sequencing techniques need to profile these separately. Can we look at them and profile them together in the same cells? And next, how are the 3D genome variations among the cell types associated with gene expression changes, right? So to answer all these questions, we developed this multiplex imaging of nucleome architecture, or MINA technique, and we imaged a whole list of a cell and the nuclear components within mouse fetal liver tissue section. First, to profile multi-scale chromatin folding, we expanded the range of genomic length scale profiled by chromatin tracing in the same experiment to over 4 orders of magnitude of genomic length. On the TET to whole chromosome scale, we traced all 50 TETs along mouse chromosome 19. And then within the TET, we actually traced at 5 kb genomic resolution the cis regulatory region of this important gene called SCD2 that has unknown architecture in its cis regulatory region in this context. And then we have, in order to do the cell type specific analysis, how the chromatin folds in different cell type, we actually profiled 137 different RNA species at a single molecule level with a multiplexed RNA fish technique. 55 of these RNA species are cell type marker genes, So we can distinguish the different cell types despite their similar morphology. And we also image the cell boundary in order to segregate the cells and get the single cell RNA expression profile. And next, to study the association between gene expression and the chromatin organization, besides the 55 cell type marker genes, the other genes are actually genes along chromosome 19. And we can basically see how the gene expression level change associated with the changes in the chromatin folding at all these length scales. And finally, to study the relationship between the chromatin folding and the other nuclear components, we actually labeled the nucleolus and the nucleus so that we can study the association of all these genomic loci with the laminar proximal region and the nucleolus. And now I'm going to quickly go through some raw data, and here's a representative trace of a whole chromosome 19, and each sphere is a tad, and there are 50 of them. And here's a representative trace of this cis regulatory region, including the promoter and the multiple enhancers upstream of the SCD2 gene. And then here is representative labeling of the whole nucleus and the nucleolus, and the computer abstraction of the laminar proximal region and the nucleoli region. And here, to image the 137 RNA species, we used this technique termed MERFISH, or multiplexed area robust fish. So it's another technique I co-developed back when I was a postdoc with colleagues. And this is the representative image of applying this technique to mouse fetal liver. And each dot here is actually a single molecule, and they are color-coded to show their gene identity, and there are 137 different genes or different RNA species that are shown on this field of view. And we can label the cell boundary also and segregate the cells. So with these single cell RNA profiles, we can easily distinguish the different fetal liver cell types. We applied a computational workflow that's routinely used in single-cell RNA-seq analysis. And here you see the t-SNE plot, and the color-coded patches corresponds to the different cell types. And we can identify which cell type is which based on the marker gene expression. That SCD2 gene, which is crucial for fetal liver development and the lipid metabolism, previously we already know it should be enriched in hepatocyte, and indeed we see from our data it's enriched in hepatocyte. And then upstream of that SCD2 gene, there are several enhancers, annotated enhancers. Which one of them actually interact with the SCD2 promoter, we actually don't know. Like in hepatocyte, it's unknown in the fetal liver context. With our technique, we set out to test that, and we basically plotted the mean spatial distances of the pairwise genomic loci upstream of the SCD2 that we profiled and imaged. And we compared such a matrix between the hepatocyte and all the non-hepatocyte cells. And we found that in hepatocyte, the distance between locus 16 and the locus 19 becomes smaller than in the non-hepatocytes. And the locus 16 is actually corresponding to one of the annotated enhancer upstream of the SCD2 promoter. And we also defined distance threshold. So we can say that distance shorter than this threshold, we see that two chromatin loci are contacting, and larger than this distance threshold, they are not contacting. If we do it that way, we also see that the contact frequency between locus 16 and locus 19 indeed is enriched in hepatocyte over non-hepatocyte. So we basically show that the MINA can reveal the fine-scale chromatin folding and shows a specific cell type-specific promoter enhancer interaction in fetal liver hepatocytes. Next, we asked about the larger scale chromatin organization, particularly the AB compartmentalization schemes in the different fetal liver cell types. And again, we did the compartment analysis for different cell types and plot their compartment scores for all the 50 TADs. And we see the compartmentalization profile between the cell types, they do differ. There's a lot of changes in the compartment scores or even switches of compartments. However, despite all these differences in each cell type, the AB compartments are stably organized in individual copies of chromosomes, and they're positioned in this polarized side-by-side kind of fashion. And these compartment score changes, they do reflect the gene expression changes, because if you have a gene that's significantly increasing its expression, it's much more likely the corresponding gene region actually have an increase in compartment score. So it becomes more compartment A-like, or switches between compartment compartment B to compartment A. However, there are also a lot of compartment score changes that are not really associated with local gene expression changes. So this seems to show that the AB compartmentalization schemes do differ in a cell type specific manner in fetal liver, and their relationship with gene expression is such that it's associated with gene expression. The AB compartment score is associated with gene expression, but it's not solely determining gene expression. And next, we asked about the relationship between the AB compartmentalization and the association with nuclear.
Can you wrap up in a minute or two?
Sure, yeah, definitely. With nucleolar or nuclear laminar. And here we are showing, marrying the frequency of the TADs being in spatial proximity to the nucleolus or nuclear laminar. And we define those nucleolar association ratio or laminar association ratio. And we plot them with compartment score. And first we see, like, persistently in each cell type, there is a negative correlation. So this is consistent with the fact that compartment B region with negative score of a compartment score, and also the nucleolar and laminar associated region, they are all enriched with inactive chromatin. However, we also see that between cell types, there are huge differences. For example, here between the erythroblast and the pro-erythroblast, these are very closely related cell types. Their compartment scores actually are almost identical. However, we see that one is systematically higher having in their nucleolar association and the laminar association than the other. And on the other hand, people have been asking me, so you see this polarized organization of the AB compartments. Is it because the B is associated with, is dragged by the laminar or nucleolar association? With our new data, we show that's not the case, because we can group chromosomes that are not associated with laminar and nucleolar. And we see that they still have very high polarization index, similar to the polarization index of those chromosomes that are associated with the laminar and nucleoli. So this basically means the laminar and nucleolar association are not solely determined by AB compartmentalization, nor vice versa. And finally, we have this unexpected discovery showing that if you look at, like, which TAD is more likely to be on the surface of the chromosome territory, and we call this the chromosome surface ratio, and you will find that the TADs that have strong AB compartment identities, those ones with very high compartment score or very low compartment score, they are on the surface. Those ones that are more ambiguous in their compartment identity, they are in the middle of the chromosome territory. This is the case for all cell types. This actually is different from what we expect from a phase separation model, because we can simulate a phase separation model of AB compartments, and we will see the opposite trend. However, if we further include surface interaction between the compartment A to the extra chromosomal stuff, or compartment B with extra chromosomal stuff, we can recapitulate the observed chromatin organization. So this shows a balance between intrachromosomal self-associating interactions and extra chromosomal interactions are necessary to establish the observed organization. So to wrap up, chromatin tracing allows direct spatial tracing of the 3D folding of chromatin in individual chromosomes in single cells. MINA enables multi-scale integrative nucleomic imaging in complex mammalian tissue and allows de novo discovery of a nucleomic organization in a cell type specific manner. And AB compartments stably exist in individual autosomes that are organized in a polarized fashion. Active and inactive X adopt drastically different folding and compartmentalization. In developing liver, the folding of SCD2 cis-regulator region and AB compartments show cell type specific organization schemes associated with gene expression. Laminar or nucleolar associations of genomic regions are correlated but not solely determined by AB compartmentalization, nor vice versa. TADs with strong AB compartment identities tends to localize to chromosome surface, and a balance between intrachromosomal self-associating interactions and extrachromosomal interactions are necessary to establish the observed chromatin organization. Finally, I'd like to thank my postdoctoral advisor, Professor Xiaowei Zhuang, and my former colleagues and collaborators for their contribution to chromatin tracing and MERFISH. and thank all the authors on the MINA work. This work was led by two talented graduate students, Miao Liu and Yanfeng Lu, and we collaborated with my collaborator, Professor Sam Katz from the Pathology Department of Yale School of Medicine. I thank my collaborator as well, and thank my funding agencies. And thank you all for attending this talk.
Well, thank you, Stephen, so much for this great talk. You were very efficient at the start and didn't give me a chance to mention all the accolades of your impressive career, but I think people will be sufficiently impressed by the talk you just gave.
Thank you.
So I'd like to start off with a question that was asked by Lavanya Ear. So she talked about the chromatin tracing stuff in the 2016 science study. Is there a reason why you did not use combinatorial barcoding and use only this sequential hybridization? And she adds, could it be because DNA fish requires way more primary probes compared to RNA fish to generate a good signal?
Great, that's a great question. In order to read out the combinatorial barcode, one actually requires the two genomic loci with different barcodes do not spatially overlap. Because if they spatially overlap, then their barcodes are essentially merged with each other in space. And through the repeated imaging, you see both of the barcodes will basically show up on the same spot, and then it's hard to read out which of the digit belongs to which genomic loci. So that's one intrinsic difficulty. I'm not saying it's not solvable, it's potentially solvable. However, in the first demonstration of the technique, we basically want to avoid this kind of complexity, because we know, well, we're looking at, contacts, regions that should be far apart, but expect to be far apart, but they actually come together. And that happens quite a bit in chromatin folding. So that's why we didn't use combinatorial barcoding back then.
Thank you. Next question is from Natalia Koganova, and I'm unmuting her now.
Hi, Stephen. Great talk.
Thank you, Natalia.
I wanted to ask you about the intermixing of the inactive X. So basically, you showed this intermixing, and what you also showed is that A and B compartments don't strictly correlate with gene expression. So I wanted to ask you, do you see this pattern of intermixing in other inactive compartments, or it is just something specific to X? In other words, can it kind of contribute to the universal mechanism of compartmentalization?
Great question. So that level of intermixing shown by basically the folding scale analysis between the spatial distance versus genomic distance, it's actually quite peculiar. And in XI, we see this. And in another situation that is known, that people have observed this, actually is in polycomb regions. For example, in Alistair Boettger's 2016 Nature paper using storm technique to look at the size of the polycomb domains in Drosophila cells. And they basically can measure the whole volume of those domains versus the genomic size of those domains, right? And you can actually convert the scaling factor of that to comparable to the scaling factor of the way that I measured it. And if you do this conversion, you will find those polycomb domains, those are small genomic domains in Drosophila. It shows the same kind of scaling as the whole X chromosome, large chromatin region in human cells. And both of these regions, they are...
Polycomb. What is interesting, the X is also polycomb, right?
Yes, right. And both of them are polycomb, enriched. So maybe that is the mechanism. So yeah, polycomb actually promotes this long distance interaction between chromatin regions, right? So you basically have some cross-linker that's very sticky in these regions, and maybe that's why they show this very special scaling.
Okay, thanks.
Next, I'll unmute Ellen Underhill. And I just wanted to mention for the people who didn't get their questions to Rob answered, he's been great and he's actually typed out the answers in the Q&A box, so you can find those there. Ellen, go ahead.
That's beautiful work. I mean, I was really excited that you were looking at the biophysical aspect with the polymer models towards the end. So do you have conditions where that polarization basically disappears?
Great question. So in the model, we actually tested 3 situations. So first one is we only have self-associating interactions among the B regions. So a B region can interact with a B region, but A regions don't really interact with A or B regions. They're just random, like bumping into each other or diffusing. And in that case, we actually see the A will wrap around the B. So the polarization index becomes much lower. It's actually comparable or even lower than the randomization control. It's becoming like A wrapping around B rather than polarized side by side kind of fashion. And in order to have the polarized organization, we do have to have AA also interact with each other as well as BB, and then we have this polarized organization.
Okay.
Thank you.
Thanks.
And then as the final question, I will read up a question from Jason Fan. He asked, is the strong AB compartment on the surface of a chromosome more likely to contact their corresponding compartment in other chromosomes?
Indeed, indeed, indeed. So even in our simulation, we actually didn't specify these extra chromosomal interaction between A to extra chromosome stuff or between B to extra chromosome stuff, what they are. They could actually well be inter-chromosomal interactions, and we are further investigating into that. So you can imagine maybe like the other strong regions from other chromosomes, they're dragging these regions to the surface of the chromosome territory.
Very cool. Well, thank you, Stephen, for your fantastic talk.